I'll start with my session. So I'll share the screen. Are you able to see my screen? Are my slides visible? Pranab, can you make sure that my slides are visible.
No, ma'am.
The slides are not visible. Okay, I'll again share my screen. Now are my slides visible?
Yes, ma'am.
Okay. Okay, so this is the revision session for this course. So if you have any doubt, you can ask me like before we start the session. So if you don't have any doubt, so what I have done for today's lecture is I have gone through the entire weekly session and jot out some videos on the important concepts. So the basic of the concepts as well as the main important concept. So we will go through each of the concept one by one. So before that, if you have any your specific doubt, you can ask. So this course molecular biology is being taken by Professor Vishal Trivedi. He is professor in Department of Biosciences and Bioengineering in IIT Guwahati. And I am Pooja Yadav. I am a PhD scholar and a PMRF fellow in Department of Biosciences and Bioengineering. So in IIT Bombay, and I am a PhD student in molecular virology lab. So we'll begin with our session. So if do you have any specific doubts, Pranam? So if you have any specific doubt, we can like discuss before going forward. Okay, so if you don't have a specific doubt, let's begin with our session. So as I told you already, so I have jotted down videos lecture, particularly from the YouTube. So I have jotted down the most important concept for the lectures, the concept that were covered in the lectures. So we will go through each of the videos one by one. So if you have any doubt regarding any concept or like you can ask in between the session or at the end of the session as you wish. So it would be probably nice if you ask in between the session only, so don't wait till the end. So let's begin. So this course, the molecular virology course, was first started with the introduction of the prokaryotic and the eukaryotic cell. So let's look into what are the main difference of the prokaryotic and the eukaryotic cell. So the prokaryotic and the eukaryotic cells are quite different in their structural as well as the organelle they have and their basic, so and the machinery that are involved in the replication, transcription and translation of the genome in prokaryotic and the eukaryotic differ a lot. So let's look at each of these things step by step. So again, what I'll need to again stop sharing my screen again and again and have to reshare the Chrome tab. So please bear with me.
Here to create a prose-based visual montage.
Those are audible.
Cells are the smallest living units of an organism. All cells have three no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment. Cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts.
Pranav, can you confirm whether these videos are audible?
Yes, ma'am. Audible. Please.
Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplast, both plant and animal cells have mitochondria.
I hope you are clear with the prokaryotic and the eukaryotic cell and function of each of the organelle that these cells have. If you have any doubt, you can ask. So next were the topic of the bacterium, and so bacteria are divided into two categories. One is gram positive and one is gram negative. So gram positive, the name gram positive and the gram negative came from the staining, which is a gram staining. The bacteria which are able to take up the gram staining are termed as gram positive, while the bacteria which are not able to take up the gram staining are the gram negative. So what are the specific features of the gram positive and the gram negative which makes them gram positive and the gram negative? So let's also look into this next video. So we will be more clear about it.
Bacteria have cell walls made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less concentrated solution to a more concentrated one, equalizing the concentration on either side of the membrane. Nearly all bacterial cell walls have a peptide polysaccharide layer called peptidoglycan, also known as murine. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram positive or gram negative, based on the chemical and physical properties of their cell walls. Gram positive bacteria
You cannot see the screen, okay? I'll again reshare the screen. Just are you able to see now, Ranam?
Yes, ma'am.
Okay, so I'll restart this video.
Bacteria have cell walls made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variations in osmotic pressure due to the different solutions they encounter, and it is their cell walls which prevent them from shrinking or swelling. As a reminder, osmosis is the process by which solvent molecules pass through a semi-permeable membrane from a less concentrated solution to a more concentrated one, equalizing the concentration on either side of the membrane. Nearly all bacterial cell walls have a peptide polysaccharide layer called peptidoglycan, also known as murine. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram positive or gram negative, based on the chemical and physical properties of their cell walls. Gram-positive bacteria have a thick cell wall, which consists of up to around 30 layers of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram-negative bacteria have a much thinner cell wall, consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membranes called diderms. We can differentiate between gram-positive and gram-negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then, a counterstain is added, for example, safranin or fuchsine. Gram-positive bacteria will retain the crystal violet dye and remain purple, while the gram-negative bacteria will be stained pink. Note that gram-positive bacteria also pick up the pink color of the counterstain. However, this is not visible when they are dyed with the darker purple color of the crystal violet stain. The reason for these staining differences is due to differences in cell wall structure, which is the chief difference between gram-positive and gram-negative bacteria. The gram stain detects peptidoglycan, and since gram-positive bacteria have a thick multi-layered peptidoglycan layer, they retain the crystal violet dye. Gram-negative bacteria do not retain the dye for two reasons. They have an outer membrane getting in the way of the crystal violet, and they lack peptidoglycan to retain the stain. Although both gram-positive and gram-negative bacteria can be pathogenic, gram-negative bacteria are more resistant to antibodies because of their impenetrable cell wall. Unfortunately, these bacteria also develop resistance more quickly. Not all bacteria can be reliably classified through gram staining. Acid-fast bacteria and gram-variable bacteria, for example, do not respond to gram staining. Acid-fast bacteria are bacteria whose cell walls retain stains particularly well. Although they aren't closely related to gram-positive bacteria, they can appear purple after the gram stain test. Gram-variable bacteria show a mix of pink and purple cells when stained. If you like this video, like and subscribe.
So I hope you are clear with the difference between the gram-positive and the gram-negative bacteria. So if you have any doubt, you can ask. Are you clear upon it, Pranav?
Ma'am,
Yes.
gram variable bacteria is not. What is it?
Gram-variable bacteria, actually these are sort of very rare bacteria which are found in nature, which are not confirmed by the gram staining. They sometimes shows gram positive, sometimes shows gram negative. So you cannot judge based on the gram positive, and they are different. Their structure are completely different than that of the gram positive and the gram negative bacteria. So there are not just gram positive and the gram negative bacteria, there are different bacteria as well. So you need to confirm by other ways, as like 16S RNA, if they are gram negative or the gram positive bacteria.
Okay, ma'am.
So let's move to our next question. So then we have a video on we have video on euchromatin and heterochromatin. Let's look into it. So this is relevantly a very important topic. So let's
भैया 50 के बाद न्यूमोकॉकल बीमारी का खतरा 5 गुना बढ़ जाता है। तो अपने और अपने परिवार के लिए आज ही अपने डॉक्टर से न्यूमोकॉकल वैक्सीनेशन की सलाह जरूर लें।
In this video,
are you able to see the video for euchromatin and heterochromatin, Pranam?
Yes, ma'am.
Okay, so I'll do it.
we'll talk about the differences between euchromatin and heterochromatin. So the building block of chromatin is nucleosome, which comprises of DNA and histones. Now, if we look into a nucleus, we would find several chromatin domain which are densely packed, and there are certain domains which are loosely packed. The densely packed regions are known as heterochromatin, whereas euchromatin is the region which are loosely packed and more accessible. Heterochromatin and euchromatin are widely different. We would try to understand how they are different at a molecular level. So obviously, from a structural point of view, heterochromatin is densely packed and euchromatin is loosely packed. Heterochromatin is associated with silence genes, whereas euchromatin is a hub for active transcription. So let's try to understand what are the molecular factors that differentiates these two structural domains in a chromatin. First of all, we need to understand the status of different histone modification along the chromatin region. Second of all, we need to understand about different histone variants. And thirdly, we need to understand about DNA methylation status. All of these factors would dictate how accessible a chromatin is and how much that can influence a transcription or any other kind of gene expression process. So first thing is histone modification. So when it comes to euchromatin, there are several histone modifications such as acetylation, and especially acetylation in H3 or H4 residues are associated with a euchromatin or an open chromatin state. Acetylations like H3K9 acetylation, H3K14 acetylation, H4K acetylations are very prominent among others. Methylation like H3K36 methylation, H3K4 dimethylation is very common in euchromatin. Phosphorylation like H3S10P is very common in euchromatin region. Heterochromatin regions are often associated with several methylations such as H3K27 dimethylation or monomethylation, H420 monomethylation, etc. So what we can appreciate at this point that the histone marks at the euchromatin region and heterochromatin regions are broadly different. And all these marks would dictate how the chromatin is tightly wrapped around in a particular region. Because if there is acetylation, you can understand the chromatin would be loosely packed and it would be accessible towards many factors. If you want to learn more about these histone modification processes, you can click on the I button. Now there are several histone variants which can delineate between euchromatin and heterochromatin regions. In euchromatin region, one can find H2AZ or H3.3 histone variants, whereas in heterochromatin region, one can find H3.1, 3.2, SENP-A, macro H2A, these kind of variants. H1 histone is another important player which can discriminate between euchromatin and heterochromatin. Often it was noticed that H1 histone deposition was fairly low in the euchromatin region. So H1 histone's job is to make the chromatin further compact. So if we have less H1 histone deposition, the chromatin is expected to be more accessible and loosely wrapped. Whereas in heterochromatin region, there is a high density of H1 histone, which can be associated with several complexes that recruit factors for heterochromatinization. Now let's look at the methylation mark and how they are different between euchromatin and heterochromatin. So in the heterochromatin region, one can see hypermethylation. So there are so many methyl marks in the heterochromatin region compared to euchromatin region, which is generally hypomethylated. Now all these factors are really important for gene expression, because in order for gene expression to happen, transcription factors need to gain access to a promoter region and thereby starting the transcription. And this accessibility issue can be solved only when you don't have too much of nucleosome density. So chromatin accessibility is a very important factor that dictates the transcription. That is why euchromatin is generally transcriptionally active, whereas heterochromatin is transcriptionally silent. Now, how to understand a particular region is euchromatin or heterochromatin like? So is there a high throughput technology to do that? Yes, exactly. By performing ATAC sequencing, we can understand for a particular cell type at a particular time which regions of the genome are heterochromatinized versus euchromatinized. So if the genome is more accessible, so we would be seeing ATAC peaks. Now, if you want a more detailed video on ATAC-seq, you can click on the I button. But if the region is heterochromatinized and the nucleosome density is very high, ATAC peaks would not be seen. That means ATAC peak would tell us about the accessibility of the chromatin at that particular region. So in this video, we looked at the differences between euchromatin and heterochromatin from different points of view, like histone variants, histone modification, linker histone, DNA methylation status, and the overall accessibility of chromatin. So I hope this was useful. If you like this video,
So basically, euchromatin is transcriptionally active because the promoter region is more accessible in the euchromatin because it is loosely packed. Why it is loosely packed? Because of the modification in the region found, like it is more acylated than that of the methylated. While in case of heterochromatin, the chromosome is very densely packed and they are more methylated when we compare to the euchromatin. So this is making the heterochromatin very densely packed, and the promoter region is not accessible for the transcription factor. So we need the accessible promoter region for the gene expression. So if this promoter region is not accessible due to the dense packing, so the heterochromatin, it will be transcriptionally inactive. So that was the gist of the video. So, and why do we need this packing? Because DNA is comparatively larger in size, so we need the compaction of DNA in order it can fit in the nucleosome. So the our DNA is around 2 meter long. So you can imagine, if the DNA is 2 meter long and cell is so small, how can this 2 meter DNA can be packed in a nucleus, not even in a cell? So for this we need to condense the DNA in so small so that it can go and incorporate into the nucleus. So how this is done? So this nucleosome, they form this nucleosome like structure. So this DNA gets wrapped around those histone protein, and they form the very dense chromatin structure, which again form the chromosome, and which stays inside the nucleus. So I hope this is clear. So if you have any doubt, you can ask. So if you don't have any doubt, so let's move to our next. So is there any specific topic you want to learn? Because we have very limited time, 2 hours. So I have lots of videos there. If you have any specific lectures or specific topic you want to like go through first, so you can tell me your choice. So I'll share my screen. Hello, am I audible, Pranam?
Yes, ma'am.
Okay, so I think I have somehow. Can you see my slides? Hello Pranav, can you see my slides? Hello, am I audible? So I have uh, videos on too many topic. If you have any specific topic you want to go first, we can go through that, or we can go through serially, but we have very limited time. So I don't think so we can go through each of the topic today. So if you have any specific topic, we can go through it first.
Okay, ma'am.
Okay, so okay, what I'll do is I'll go through the my each slide. Then if you find any topic interesting, let's go through the slide once, and if you want to know about the topic more, then we'll go through that topic, or either we'll go through the topic one by one. So after euchromatin and heterochromatin, I had electron transport chain, like for ATP synthesis in the cell, how mitochondria, so ATP synthesis occur in eukaryotic cells and the prokaryotic cells. Then we have biomolecule breakdown to produce the energy. So one is the glycolysis pathway, the other one is the Krebs cycle, and then the beta oxidation. Then we have apoptosis.
Ma'am, beta oxidation.
Yes, we'll go through the beta oxidation.
Ma'am, beta oxidation.
Yes, okay.
Hey, still thinking about joining the 30-day Python using AI bootcamp by AI for Techies? So what if in 30 days...
This video is visible and audible?
Yes, ma'am.
Okay.
In previous videos, we've learned that we can take glucose and other carbohydrates and oxidize them through glycolysis and the Krebs cycle. And we know when we take carbohydrates like glucose and oxidize them through glycolysis and the Krebs cycle, we create these reduced cofactors. And we know these reduced cofactors can be used to fuel the electron transfer chain to create ATP. So that's the way we can take carbohydrates and oxidize them to create ATP. But we can do the same thing with free fatty acids. We can take this free fatty acid and oxidize it to create ATP through a process known as beta oxidation. So the way we do this is we take the free fatty acid and we go through one round of beta oxidation. And with each round of beta oxidation, we create one FADH2 and one NADH. So then we do another round of beta oxidation and we create more of these reduced cofactors. Then we do another round and create more of these reduced cofactors. And something important to realize is with each round of beta oxidation, we lose carbons. We lose 2 carbons in the form of acetyl-CoA. Then we do another round of beta oxidation and lose 2 more carbons in the form of acetyl-CoA. But the point is, as we keep on doing beta oxidation, rounds of beta oxidation, oxidizing this free fatty acid, as we keep doing beta oxidation, we create these reduced cofactors, which can fuel the electron transfer chain to create ATP. So that's a way we can oxidize fat to create ATP. And if you're curious on the exact mechanism, first we take the free fatty acid and we activate it by reacting it with an ATP and a CoA. But essentially we activate it, forming this compound. Now we're ready to go through beta oxidation to create ATP. So what's the first step? First, we see these hydrogens. We essentially react them with one of these FADs. So this FAD reacts, stealing those hydrogens and those electrons. And when we do that, we produce an FADH2. And as we stole those electrons, we created this double bond. So now we've created one of these reduced cofactors. So that's the first step. Now we just do a simple addition reaction where we have this double bond, so we can simply add a water through an addition reaction. And again, this is just basic organic chemistry, where again we know these pi bonds attack the hydrogen, then we create a carbocation where this oxygen attacks. But the point is, it's just a simple addition, and now we have a hydroxyl. Now, once we have the hydroxyl, we can use one of these NADs to react, essentially stealing more hydrogens. And when this NAD steals more hydrogens, it produces an FADH. And as we stole the hydrogens, we've created one of these carbonyls. But now we've created our second reduced cofactor. So that's good. So now we've created our reduced cofactors. But now what do we do? Well, now we take one of these CoAs, and essentially we go through a reaction where we deprotonate this sulfur. So now we have the sulfur nucleophile, and we essentially go through a reaction where we form a bond and we break a bond where these electrons fall in this carbon. And the exact mechanism is, again, we nucleophilically attack. When we attack, we form a bond, and once we form that bond, we push these pi electrons up on the oxygen. So when we do that, we form this tetrahedral intermediate. We formed a bond and pushed those electrons up. Now what happens is the electrons scooch back down, forming a double bond, and when that happens, now this bond breaks and these electrons fall in the carbon. And when we do that, we form this structure. So we've done it. Remember what we were doing. we form a bond and break a bond, and these electrons fall on the carbon. So we form a bond and break a bond. And then again, this carbon would get protonated, and now we've done it. So now we've gone through a round of beta oxidation. Remember, we've created these reduced cofactors, and we lost carbons in the form of acetyl-CoA, so that we've gone through a round of beta oxidation. And now we can go through another round, losing more carbons of acetyl-CoA and creating more reduced cofactors. And then we do another round, and etc. And we keep on doing rounds of beta oxidation, keep on losing carbons and creating these reduced cofactors. So as we go through beta oxidation and oxidize this fatty acid, we create these reduced cofactors, which we know fuel the electron transport chain and can be used to create ATP. So this is a way we can take fat, oxidize it to create ATP. And if you're curious on the context of when our body's doing beta oxidation, well, we know in our bloodstream, we have glucose in our bloodstream. And it's important to make sure we have glucose in our bloodstream to keep our brain happy. Our brain requires glucose for it to function properly, and it gets its glucose from the blood. But what happens if we haven't eaten a meal for a couple of hours? Then our blood glucose concentrations drop, and that's dangerous, and now that's dangerous for the brain because now it doesn't have a source of energy. So essentially what happens is the pancreas senses that the glucose concentrations have dropped, and that's bad. So in response, it releases glucagon. That glucagon tells the liver to go through gluconeogenesis, to use non-carbohydrate precursors as a source of carbons to create glucose molecules. So glucagon tells the liver to go through gluconeogenesis to biosynthesize these glucose molecules, so now we can dump those glucose molecules in the bloodstream to keep the brain happy. But this process of gluconeogenesis requires a lot of ATP. This is an anabolic process which requires a lot of ATP. Where do we get that ATP? Well, again, that glucagon also tells the adipocytes to hydrolyze these bonds, go through a process known as lipolysis, to essentially hydrolyze these ester bonds to release those free fatty acids, which can be sent to the liver mitochondria. Now, when those free fatty acids enter the mitochondria in the liver, the hepatocytes in the liver, now those free fatty acids now this is when beta oxidation occurs. And again, glucagon also activates the beta oxidation, but now the beta oxidation occurs to create these reduced cofactors to fuel the electron transport chain to create ATP to fuel gluconeogenesis. So that's the context of when we do beta oxidation. We burn these fats and go through beta oxidation to create ATP to fuel gluconeogenesis to biosynthesize glucose molecules so we can dump it into the bloodstream to keep the brain happy.
So are you clear on the beta oxidation? So in order to so beta oxidation is again a kind of energy producing step where the long chain fatty acids are used to create FADH, FADH2 and NADH, then which are later on used in the electron transport chain to produce the ATP for the requirement. So first the long chain fatty acid, they turn into fatty acyl coenzyme. So these two carbon at each cycle, these two carbons are removed. So at the end of each cycle, we are left with 2 molecules of acetyl coenzyme. So this is how the reactions take place, and at each of the cycle we get FADH2 and NADH. So that's how the beta oxidation takes place. So let's move to our next slide. So the next slide was on apoptosis. So apoptosis is cell death. Are you able to see the slides? Are my slides changing on the screen?
Yes, ma'am.
Okay, so the next, so do you want me to go through the apoptosis, or is it clear to you? Okay, then we have the video on DNA as a biomolecule. protein, biomolecule, then cell cycle, mitosis and meiosis, then how DNA as a genetic material was identified by Hershey and Chase experiment, then the central dogma, the slide on the central dogma, how DNA is first replicated and then RNA is formed via the process of. Okay, is my slide visible or not? I think something has happened. Hello Pranav, I actually lost the connectivity through my main account. I am trying to join in again. I'm sorry about that. So are you able to see my screen?
Yes, ma'am.
Okay, so uh, we had gone through beta oxidation. So the next is apoptosis. So do you want me to play the video on the apoptosis, or we can go ahead?
Ma'am, another one.
Okay, so the next video was on DNA as a biomolecule, then protein as a biomolecule, then the absorption at which the protein and nucleotide, the nucleic acid, they show absorption, on what are the wavelengths on which they show absorption. So the nucleic acid, they show absorption at 260 nanometer, while protein they show at 280 nanometer. So then we have cell cycle, mitosis and meiosis. Then Hershey and Chase, how they confirmed that DNA is the genetic
Ma'am,
material.
this one
Yeah, so we can go through this.
Hershey and
Yes.
Chase
Okay, we'll do this.
experiment.
Yes, sure.
नेचुरल लुकिंग कलर का भरोसेमंद सीक्रेट, इंडिया का सबसे भरोसेमंद है कलर गार्नियर ब्लैक। Naturals.
A bacteriophage is a virus that infects a bacterium. With the help of bacteriophages, Alfred Hershey and Martha Chase in 1952 performed an experiment to demonstrate that DNA was the genetic material. Hershey and Chase grew some viruses in a medium that contained radioactive phosphorus. Viruses grown on this medium contained radioactive DNA. Similarly, they grew some viruses in medium containing radioactive sulfur. Viruses grown on this medium contained radioactive protein. These two radioactive viruses were allowed to infect bacteria separately. After infection, the viral coats were removed from the bacteria by agitation. And the viral particles were separated from the bacteria by using a centrifuge. Finally, the bacteria were tested for traces of radioactivity. The bacteria infected with the virus having radioactive phosphorus contained radioactive DNA. This indicates that DNA was passed to the bacteria from that virus. The bacteria infected by the virus having radioactive sulfur consisted of normal sulfur. This indicates that the proteins were not passed from the viruses to the bacteria. With this, they concluded that DNA was the genetic material that was passed from virus to bacteria.
So in Hershey and Chase experiment, what they did, so they had bacteriophage. So the bacteriophages are the viruses that infects the bacteria. So they radio label the bacteriophages. So in one culture they radio label the bacteriophages with radioactive sulfur. So sulfur are found in the protein. So these sulfur go and attach to the capsid of the virus. And in another media they added radioactive phosphor. And in DNA phosphate is present. The phosphate group is present. So this radioactive phosphate is being taken by the DNA of the virus. So after the infection, so how the virus infect, so first they go and attach to their host, and then they inject their genome inside the host cell. So once they have injected the genome in the host cell, the scientists, they removed the attached empty phage particle with the help of the centrifugation. So then they looked for the radioactivity present in the host cell. So and then they found that the DNA, the new DNA, the radioactivity was found in the DNA, and in the case where the DNA was radio labeled with the phosphate group. But not in case of where the phage was radio labeled with sulfur. So that's how they confirmed that it was the DNA that got transferred from the virus to the bacteria. let's look into the replication of difference between the prokaryotic and the eukaryotic DNA replication.
I have 12 emails, 30 messages, and a proposal due by 10. Now I'm already underwater.
I'm in the same boat. In this video, we'll be discussing about overview of DNA replication, and particularly we'll be more focused towards the enzymes and proteins involved in the replication process of prokaryotes and eukaryotes. In DNA replication, the single DNA molecule having two strands is replicated to form two DNA molecules. We see we are having a DNA molecule. Its two strands acts as parent strands to make two DNA copies. So on these parent strands, the enzymes and proteins will work on to...
Hello?
Yes, ma'am. Ma'am, network is problem, so...
Oh, it's okay. It's alright. So, okay, then I'll again reshare. Were you able to understood the Hershey and Chase experiment?
Yes, ma'am.
Okay. It's alright. I know the network issue is there everywhere, so I understand. So, after Hershey and Chase experiment, so this is the central dogma, like how the DNA self-replicate and then the transcription leads to RNA synthesis and from the DNA, and then the translation of protein from the RNA. So this is a normal video on the DNA replication. Then the difference between the prokaryotic and the eukaryotic DNA replication. So please, Pranab, interrupt me which specific video you want to look into more detail. I'll just go through each slide one by one. So then we have the end replication problem. So especially in the eukaryotic replication. So then we have prokaryotic transcription. Yes?
Ma'am, telomere replication.
Yeah,
the previous slide.
Yes, we will go through this.
Audio jungle.
So are you able to see this? Pranab, are you able to see the video?
Yes, ma'am.
Okay,
In our earlier videos, we have already discussed how DNA synthesis occurs in prokaryotes. But a unique problem arises when the replication fork reaches at the end of the chromosome. The enzyme DNA polymerase can only elongate from a free 3' hydroxyl group. RNA primers provide free 3' hydroxyl group at regular intervals along the lagging strand. However, the leading strand elongates continuously till the end of the chromosome. Even if the final primer provides a 3' hydroxyl group at the end of the chromosome, the end of the lagging strand can't be completed. Because once the final RNA primer is removed by the DNA polymerase I, there is no free hydroxyl group at the end where DNA polymerase can synthesize a new DNA. These gaps at the end of the lagging strand must be filled by a special mechanism to prevent the chromosome ends from shortening with each round of cell division. Eukaryotes have their long repetitive nucleotide sequences at the end of their chromosomes, which is termed as telomeres. These telomeric DNA sequences contain a guanine-rich series of repeats which is recognized by the telomerase. The telomerase carries a short piece of RNA template complementary to the DNA repeat sequence, acts as template for telomere DNA synthesis. Telomerase binds and elongates the ends of the parental strand by adding more copies of repeated sequence in 5' to 3' direction as it moves down the parental strand. This is termed as RNA templated DNA synthesis, which occurs via reverse transcription mechanism. After that, the lagging strand is completed by DNA polymerase alpha, which have DNA primase as one of its subunits. Thus, no information is left to be copied from the parental DNA to the new DNA. In this way, telomeric replication occurs to prevent eukaryotic chromosome from shortening with each cycle of cell division.
So this was the end replication problem and how it was solved. So first the telomerase, they have this RNA sequence, which with the help of this RNA sequence, so with the help of the reverse transcriptase, so the DNA is synthesized, and then the DNA polymerase alpha, it has the primer sequence of that newly synthesized DNA, so then it extends the polymerization of the single stranded DNA that is left behind. So I hope this is clear.
Yes, ma'am.
Okay, so then we have prokaryotic transcription, eukaryotic transcription, then we have post-transcriptional modification, then gene regulation.
Ma'am.
Gene regulation.
Ma'am.
Yes. Post-transcriptional.
Post-transcriptional modification.
Yes. Okay, we'll go through this.
Thane's biggest developer is now in Mumbai. Welcome to the address by GS Bandra by Raymond.
So are you able to see the slides or see the video?
Yes, ma'am.
Okay.
Messenger RNA or mRNA is a type of RNA that carries genetic information for making proteins. It has three main parts: a 5' untranslated region 5' UTR at the beginning, a protein coding region in the middle, and a 3' untranslated region, the 3' UTR at the end. In prokaryotes, the mRNA is ready for translation as soon as it is transcribed from DNA. There is no need for any further modifications. Prokaryotes do not have a nucleus, so the mRNA can be translated by the ribosome in the cytoplasm while it is still being transcribed by the RNA polymerase. This means that the transcription and translation happen at the same time in prokaryotes. In eukaryotes, there is a nucleus, so transcription and translation are separated. Initial RNA transcript is called the pre-mRNA, which needs to be processed in the nucleus before it becomes the mature mRNA. The first step in processing the pre-mRNA in eukaryotes is adding a 5' cap. This is a modified guanine nucleotide that is attached to the 5' end of the transcript with a special 5' to 5' bond. Then some methyl groups are added to the cap and the next two nucleotides. The cap helps the ribosome to recognize and bind to the mRNA in the cytoplasm. The last step in processing the pre-mRNA in eukaryotes is adding a 3' poly-A tail. This is a string of adenine nucleotides that is added to the 3' end of the transcript. There is no specific sequence that signals the end of the gene, and sometimes RNA polymerase may transcribe beyond the gene. There is a sequence AAUAAA, the pre-mRNA that marks the end of the 3' UTR. Some proteins recognize this sequence and cut the pre-mRNA 10 to 30 nucleotides after it. Then another enzyme, poly-A polymerase, adds the poly-A tail to the cut at 3' end. The poly-A tail makes the mRNA more stable and protects it from being degraded by the enzymes in the cytoplasm. The pre-mRNA transcript also contains non-coding sequences called introns, which are interspersed with the coding sequences called exons. The introns need to be removed from the pre-mRNA. This is called splicing. Splicing is done by complexes of small nuclear RNAs or snRNAs and proteins called snRNPs or SNRPs. The SNRPs bind to the intron, cut its left end from the upstream exon, loop the intron back, cut its right end from the downstream exon, and join the two exons together. The result is the mature mRNA without introns. After the pre-mRNA is processed, the mature mRNA leaves the nucleus and enters the cytoplasm. There, it is recognized by the ribosomes, which start to translate it into a protein.
So I hope the post-transcriptional modification is clear. Pranab?
Yes, ma'am.
Okay. So we then have gene regulation, prokaryotic translation, eukaryotic translation. Then we have some techniques, laboratory techniques. The first is plasmid DNA isolation, southern blot, western blot.
Ma'am, plasmid DNA isolation.
Yes.
Plasmid DNA isolation.
Yes, sure.
Gorgeous summer skin के लिए Nivea Aloe Hydration. Skin is for being summer ready.
I hope you can see the video.
Yes, ma'am.
This video will talk about plasmid DNA isolation via the alkaline lysis method. Plasmid DNA is a really important tool in molecular biology lab. People who are cloning the gene, they need to propagate the gene in a bacteria by transformation reaction. If you want to know more about transformation, video is there in the I button or description. But anyway, propagating the plasmid, amplifying the plasmid, or storing the plasmid is really important. So overall, in this video, we'll try to understand how from bacterial cells one can chemically isolate the plasmid in a purified form. So there are two methods by which one can do that. There are column-based purification, which use the principle of affinity chromatography for purifying, and there is also chemical purification, which use fundamental chemical-based approach to purify the plasmid. Obviously, you can understand the first one is kit-based and customized, so this is more costly, whereas the second, chemical purification or alkaline lysis, is an inexpensive method. It's very cheap, but very efficient. So in this alkaline lysis method, you start with solution number 1. In a moment, I'll tell you the composition, but you first resuspend the bacterial cells by resuspension buffer. In the resuspension buffer, you have tris, EDTA. EDTA is very important because it chelates divalent metal ions such as calcium and magnesium, which are also important for DNases. So by chelating these divalent ions, you can also chelate DNase activity. Also, if you remove these calcium and magnesium, it would destabilize the membranes as well. Glucose is also added in this solution to maintain the osmolarity and ensures the cells doesn't prematurely burst. So overall, the resuspension buffer has a composition like this. So in order to prepare 100 ml of resuspension buffer or solution 1, you need to add all these components: glucose, tris-Cl, EDTA, water, and have to adjust the volume till 100 ml. Then the important part is the alkaline lysis. In order to take the plasmid out of the cell, you have to lyse the cell to take out the plasmid. And this lysis step is performed by solution number 2. This solution number 2 can be imagined as a lysis buffer, which contains sodium hydroxide and SDS. So obviously, by presence of sodium hydroxide, you can understand this particular solution is highly alkaline, and that gives us the name, the alkaline lysis. Anyway, the alkaline lysis step is performed by solution 2, whose composition is exactly mentioned here. But I'll tell you what exactly happens. After putting the alkaline lysis solution, the solution 2, you mix it gently and wait a little bit of time such that the lysis can happen. In this step, first of all, NaOH loosens up the cell wall, SDS pops hole in the membrane, and that releases the overall chromosome of bacteria and also the plasmid DNA in the solution. Now, also the DNA gets denatured because all the interaction between the DNAs, the base pairs, the hydrogen bonds are now disrupted because there is an alteration in pH in the solution, right? And this is not favorable for hydrogen bond formation because it is highly alkaline. So DNA gets denatured. Particular point of time, if you take the solution and open the tube, you would see strand-like appearances coming out of the tube. Now the next step is neutralization. This extremely alkaline environment has to be neutralized. And this neutralization is done by potassium acetate and acetic acid. Obviously, by putting acid, you can neutralize the base. So it forms a salt known as potassium dodecyl sulfate with the SDS, which is present in the alkaline lysis solution. And this is an insoluble salt. So after putting this solution 3, what happens is you see curdy precipitate like stuff appearing in the tube, which are highly insoluble. You can visually see them. So then you wait some time and then centrifuge them. Now, once you add the neutralization buffer, the waiting is important because you want to wait for precisely the time that would allow the circular DNA or the plasmid to renature, but it doesn't give enough time for the denatured cellular DNA or the chromosomal DNA to renature. So you have to ensure that, okay, renaturation happens only for the plasmid DNA. So the timing is critical in this step. After that, one should centrifuge them and precipitate these insoluble substances, the salt of dodecyl sulfate. So after that, what happens? The aqueous portion is taken out, and along with this aqueous portion, 0.6 volume of isopropanol is added to precipitate the plasmid DNA which is supposed to be in this solution. Now, isopropanol makes the DNA unhappy. So isopropanol first forms bonds with the DNA. DNA doesn't interact with the water, so it's not anymore solubilized. It's now precipitated. After ultimately, it forms a pellet after isopropanol precipitation, and this pellet can be washed again by ethanol washing. So ethanol is added, the precipitation is again done. So ultimately, after that, the pellet is dried and resuspended in nucleus-free water, or let's say some tris buffer. This plasmid, once isolated, can be stored in -80 or -20 degrees centigrade for years. Now, how do you know the plasmid is good quality that you have isolated? So you can run those plasmid in a gel, and it should show you several bands. I mean, there are many outcomes possible, like bands correspond to nicked plasmid, linear plasmid, supercoiled, or circular, closed circular plasmid. The predominant form that you should be expecting is basically the supercoiled one, and that's why you should be getting the thickest band in the supercoiled region, and other bands should be very faint. Anyway, you can also check the quantity of plasmid, that how much is the yield of this isolation process. So one can put a 1 microliter plasmid there, and ultimately one can check the absorbance at 260 by 280 nanometer. It should come around like more than 500 nanograms per microliter for a good yield, and 260 to 80 ratio should be 1.8 for a pure DNA. If this ratio is not proper, then there could be a problem or contamination in your plasmid DNA. By looking at these parameters, you can understand how good the isolation is. You can find more flashcards and notes in my Facebook page, and you can find more.
Supercoiled band is more preferred because the supercoiled band is more intact form of the plasmid. So if we have an open plasmid or a nicked plasmid, open circular plasmid or a nicked plasmid, then there might be possible that the denaturation of the plasmid has happened, and the renaturation did not occur properly. So if the renaturation has occurred properly, it would have supercoil. So DNA has the tendency to supercoil. So that is why the supercoil band is most preferred, because the entire DNA has renatured completely. I hope this is clear. This solves your doubt. Okay. So then we have southern blot.
Ma'am,
Yes, Pranav, can you repeat?
Western blot.
Yeah, western.
Ma'am, Western blot.
Okay, we'll go on. Southern western. I hope the video is visible.
Yes, ma'am.
Okay.
When working with proteins, one cannot get around the technique called protein immunoblot, or simply the Western blot. Western blotting is a method to confirm the presence of a specific target protein in a sample. Before the analytical technique Western blot comes into play, everything starts with an SDS page. Upon treatment with SDS, all proteins in a sample are denatured and covered by negative charges. The proteins have a similar mass-to-charge ratio and travel through the gel to the positively charged anode. This allows to separate proteins according to their molecular weight. After staining the polyacrylamide gel, protein bands become visible. Marker proteins as a reference help to determine the molecular weight of proteins in the sample. This band here, for example, contains proteins with a molecular weight of approximately 60 kilodalton. However, that will not reveal which proteins are present in this sample. It could be the protein of interest, which has a molecular weight in this range, but the band could have also been caused by other proteins. To confirm the presence of a specific protein, a Western blot has to be performed. The detection of the protein of interest is based on antibodies, designed to exclusively bind epitopes of the target protein. The Western blot setup starts with the gel, containing the separated proteins. This is placed on top of a membrane, often a nitrocellulose membrane. This membrane is capable to bind large amounts of protein. Later, the idea is to transfer the proteins from the inside of the gel onto the membrane. This gel membrane construct is flanked on both sides by filter papers. All parts here are soaked in buffer to facilitate the electricity transmission later. Finally, everything is fixed between the cathode and the anode. The electric current pulls the negatively charged proteins, which are located in the gel, to the positively charged anode. This transfers the proteins onto the membrane. From now on, the membrane has to be incubated several times. The first step includes blocking. Therefore, the membrane is incubated with milk or BSA. Blocking should prevent antibodies from unspecifically binding to the membrane or to other proteins. After blocking, the membrane can be incubated with the primary antibody. This antibody should be selected to only bind to the protein of interest. After incubation with this primary antibody, a washing step will wash away the unbound antibodies while not removing the antibodies that are bound to the protein of interest. The next step involves the incubation with a secondary antibody, which has to be specific for the primary antibody. After washing, only those secondary antibodies remain which are bound to the primary antibody. The detection of the protein of interest is the last step in Western blot analysis. For that purpose, the secondary antibodies are conjugated to an enzyme. For many Western blot assays, secondary antibodies are conjugated with horseradish peroxidase. The protein of interest is bound by a primary antibody. The secondary antibody binds to the primary antibody, and when the membrane is incubated in a substrate mixture, the enzyme converts the substrate, thereby producing a chemiluminescent signal that visualizes the protein band. The presence of the target protein in these samples has been confirmed. Make sure to subscribe to stay updated for new videos, and please like this video if it was helpful to you. Thanks.
I hope now you are clear on the Western blot. So if you have any doubt, you can ask. Okay, so let's move. So then we have PCR. We have real-time PCR or quantitative PCR.
Yes, ma'am. Real.
Yes.
qPCR.
Yes.
qPCR, quantitative PCR, or real-time PCR is an effective technique to quantitatively determine the presence of a target sequence or gene in a sample. Up to date, qPCR is the gold standard method to test patients for viral infections such as COVID-19. For that, a swab is taken which may contain viral material. The RNA is extracted and reverse transcribed into DNA. The DNA can then be analyzed with qPCR in a light cycler. In conventional PCR, a DNA sequence of interest is amplified. The presence or absence of that DNA sequence is detected in an endpoint analysis after reaction completion. During quantitative PCR or real-time PCR, the accumulation of the amplification product is visualized with fluorescent reporters, and the reaction progression is monitored after each cycle. In qPCR, the product amplification is therefore observed in a quantitative perspective. With each PCR cycle, the amount of PCR product doubles, which can be detected as an increase in fluorescence. This generates a classic qPCR curve. A defined signal intensity has to be exceeded until the product is abundant enough so that the fluorescence is detectable over background. The cycle number at which enough amplified product has been accumulated to exceed that threshold is referred to as the CT value. qPCR is mainly performed using one of these two assays. In the cyber-green assay, the fluorescent reporter is an intercalating dye that binds to double-stranded DNA. The Techman qPCR method works with probes specifically designed to bind the sequence of interest. Upon synthesis of the complementary DNA strand, the probe is cleaved, which results in a fluorescent signal. In the cyber-green qPCR method, one PCR cycle starts with the separation of the two DNA strands of the amplicon. During denaturation, the primers bind to their complementary parts, flanking the sequence of interest. The polymerase starts with the synthesis. The cyber-green dye binds the double-stranded DNA fragments, and the fluorescence is monitored after each cycle. In the Techman assay, short DNA probes are provided, which are attached to a fluorescent reporter at the 5' end. The reporter activity is suppressed by the 3' quencher as long as they are in near proximity. The strands separate in the denaturation step. After this, the primers can bind, and the Techman probes are designed to specifically bind in between the target sequence of the amplicon. The TAC DNA polymerase has a 5' exonuclease activity, which will shoo away the target-specific probe. Cleavage of the Techman probe separates the reporter from the quencher, which leads to the emission of a fluorescent signal that can be detected after each cycle. In both assays, the increase of fluorescence along product amplification generates a curve. During gene expression analysis, multiple genes can be compared. The number of cycles after which the signal intensity reaches the threshold is shown as the CT value. The presence of gene 1 is detected after 21 cycles, whereas the presence of gene 2 is detected in cycle 25. In general, low CT values go along with high DNA concentrations, and high CT values mean that signals are detected late, indicating low DNA concentration. Quantitative PCR finds applications in various fields. It can be used for diagnostics to detect infections with pathogens, and it is frequently used for gene expression analysis. For more scientific content, check out the videos on this channel and make sure to subscribe to stay updated. Thanks.
भैया 50 के बाद
I hope the concept of the qPCR is clear, Pranam. Or you have any doubt, you can ask. Okay, so if it is clear. So now we have video on molecular cloning, then types of cloning vector, then screening, so until now I have a lecture video on the video till lecture 10. Then we have seen videos on lecture 11 in the previous lecture. So I'll just open the lecture 11 slide. We can just quickly go through the lecture 11 as well. So are you able to see the lecture 11? So for the lecture 11, we had video on homologous end joining. Then we had video on transposons, site specific recombination. So then we had the video on zinc finger nucleases. So all are the gene editing mechanism, either by homologous recombination and non-homologous recombination, and how these techniques are used for the genetic engineering of the genome, either for the knockout of a particular gene or for the knock-in of the particular gene, or just to modify a particular gene based on our research question or the objective. Then the video on the zinc finger domain, how the zinc finger nucleases are formed. Then we had the video on the CRISPR. So that was it for lecture 11. So in lecture 11 we learned the various way of genome editing, the advanced way of genome editing. So till now we have covered this much only. So I have a last lecture on this Tuesday, so where we will be learning of the application of the molecular cloning and the whole lot of molecular biology. So that will be our last lecture. Yes, that was all covered in the previous class. It was very recent, that is why I did not include in this video, because we had the shortage of time. So if you still want to go through some of the videos here, so I'll again go to each of my slides. We have still time. If you want to look for the few of more concept, we can go through it, or what we can do is we can end the session here. I will just quickly go through the slides, so interrupt me in between where you want to see the detail of the particular concept. So we had seen this prokaryotic and eukaryotic cells, gram positive and gram negative bacteria, the euchromatin and heterochromatin, electron transport chain, ATP synthesis, glycolysis, Krebs cycle, beta oxidation, apoptosis, DNA biomolecule, protein biomolecule, cell cycle, mitosis and meiosis, Hershey and Chase experiment, DNA replication, the difference between eukaryotic and prokaryotic DNA replication, then telomere replication, prokaryotic transcription, eukaryotic transcription, post-transcriptional modification, gene regulation, prokaryotic translation, eukaryotic translation. Then we had gone through some of the techniques like plasmid DNA isolation, southern blotting, western blotting, PCR, qPCR, molecular cloning, types of cloning vector, then how the the recombinant, the transformed or the transfected cells were screened for the recombinant DNA. So this was it for our revision session. So if you have any doubt, you can ask. Okay, so if you don't have any doubt, we can end our session here. We have our last class on Tuesday, so let's meet again on Tuesday. So thanks for joining in for today's lecture. So we can end the session here. You can leave the meeting. Thank you for joining in.